Thanks, everybody. First of all, thank you to Cure Cancer for organizing a really great event and getting us all together. I'm really excited to see everybody's projects. And so today I'd like to talk you through some of the work that we've been doing on an immune population called T cells, and they're my favorite population of all time. We're located here at the Doherty Institute in Melbourne. So I know it's probably more famous for its COVID research, but I can guarantee you there's also some awesome immunology happening as well. And so to kind of think about it from a broad perspective, our ultimate goal is to really improve cancer patient responses to radiation therapy. And this was motivated by our finding that a cancer-fighting immune cell can actually survive irradiation and still go on to protect the host. And so to introduce you to our main character, this is our killer T cells, our CD8 positive or cytotoxic T cells. I just want to take you through what they experience in their life, just to give you an overview of what they do in the context of cancer. So naive T cells, they begin as a naive state. They will encounter a foreign particle, whether it be a virus or a bacterially infected cell. But in this case, they can also recognize malignant populations, so cancer cells. They then, once they recognize a foreign particle, they then begin to expand rapidly into what's called effector T cells. And so if you were to count the number of T cells here, this is what this graph is showing, you'll see that upon this primary exposure, you kind of start off in low numbers and you get this really, really high population of T cells. It's actually this population of effector T cells that act like your soldiers. They can go out to the infected organ or the infected site and begin to eliminate that threat. And they do that in a whole variety of ways. They can secrete cytokines, they can release these sort of bullet-like molecules, so granzymes, perforin, etc. And really their whole purpose is to eliminate that threat. And so what happens when, let's say, in this case was a melanoma, you get rid of that cancerous tissue? What happens to all these effector T cells? Well, we don't need that many of them because it's quite a lot of cells, and you can only imagine over the time of a host, they keep accumulating, so you just run out of space. And so there's a very clever mechanism where they go through what's called a contraction, and about 90% of these cells actually die, but they don't just become effector T cells hanging around. They actually begin to develop into another state of T cells. And these are now called memory T cells. So again, you can see this contraction of the cell numbers. However, they've actually changed phenotypically and transcriptionally to become memory T cells. And so what is a memory T cell? You can only imagine by its name. It's a cell, a T cell that is there to patrol and remember the original threat. And its whole purpose is that once it encounters that threat, is to rapidly respond. And so if, for example, the same tissue encounters the secondary exposure of that original threat, you actually find that these memory T cells can not only act quicker, but to a greater extent. So they actually have a superior capacity to go out there and kill again. So this is really the basis of vaccination. This is what we do. It's like a dress rehearsal, a run through to generate memory T cells that make sure that when you see the threat, you're able to respond rapidly. And what's the whole point of that? The whole point is that you get a very, very quick response to prevent the progression of a disease. And at the end of the day, to protect the host. So we think that memory T cells are crucial for the durability of our immune response. And so when we think about memory T cells, their whole purpose is to constantly survey the body. And how do you do that when you have so many organs, so many tissues, a whole network of blood vessels? So we know that memory T cells can circulate through the blood and through lymph, for example. And it was thought for a very long time that, if, for example, you have an infection in the toe, you have an infection somewhere random in your body, these memory T cells will sense the threat and they can travel all the way to that site and protect the body or protect the host and fight off that infection. But in fact, what we know up until about 10 years ago, so it's relatively new actually information, is the fact that there's actually another population or a very distinct population of memory T cells, and they are called tissue-resident memory T cells, or what I'll refer to them today as TRM cells. So unlike these memory T cells that can travel through the blood and through the circulation, these tissue-resident memory T cells are the ones that are permanently embedded within your organs. They do not go anywhere. Their whole purpose is to survey your tissues. And the one thing that became very apparent as we began to study these cells is that it's not just about where they live, but their whole identity is so different from blood T cells. And so, not only the way that they look, but also the way that they behave. And in fact, we know that TRM, they can't leave the tissue, they're permanently resident in that site, but they actually can provide superior protection than the blood T cells. So there's something really, really important about these cells. And you can only imagine how critical it will be to understand these cells in the context of infection, but also in the context of cancer. And this is probably one of my favorite slides of all time because I really love this video. What we're looking at here is we're looking at our T cells, our TRM cells in the skin. These are represented in green. And they're actually interacting with melanoma cells, which are in these big blobs of red here. I'm not sure if I can actually zoom in, but I don't know, is my screen zooming in here? It's just the coolest thing ever because you can actually see their movement. They're not these like static cells. They're actually constantly surveying their environment. And what's really critical is the fact that you can see these TRM cells responding or interacting with the melanomocytes. And what is happening here is actually what's called cancer immune equilibrium. So not only can TRM cells prevent the outgrowth of cancer, or prevent the tumorigenesis rather, but also prevent this outgrowth. So it's actually keeping these tumor cells in check. So we really understand that these TRM cells are critical for the protection of solid tumors. So what happens actually if you take away TRM cells? I'm telling you they're important, but what actually happens if we were to wipe away all those green cells from this screen? What you find is that these preclinical models, which had no evidence of tumors, now begin to outgrow tumors. And so they really are keeping this all in check. And something that we found again a few years ago was that these TRM cells were not just confined to the kind of tissues, but we're also able to find them within solid cancers. And so what we're looking at here is a plot of all the T cells in a triple negative breast cancer sample. These are human samples, and every dot is a cell. And you can see that there's different flavors of T cell. Every color is a different flavor. But I think critically, we can observe that we can identify these TRM cells within the tumor itself. And I think what was the most kind of striking finding is that if you stratify patients that have more or less TRM cells, it's those patients that have higher TRM numbers that can actually do a lot better in the long run. So they have a lot better survival in both a melanoma as well as in a breast cancer as well as in a melanoma. So it really is across different cancer types. It's not just restricted to one. It's the concept that if you have more tissue T cells, you have a better survival prognosis. And so coming back to our goal and our aim, when we think about common therapies, we know, I'm sure this crowd is very familiar with the different ways for shrinking tumors. Of course, there's surgical resection of the tumor. We have chemotherapy and radiation therapy. Also, this revolutionary kind of pillar, which is the immunotherapy aspect. We're using drugs to boost the immune system. And often these types of therapies are either used alone when very effective or in combination for their synergistic effects. Something we're particularly interested in is radiation therapy. And this is the concept where you can take a solid cancer and expose it to really, really high levels of ionizing radiation. And the concept here is that the radiation itself can actually result in the DNA damage of these cells. And so you get DNA damage, the cells can no longer grow, they can't survive, they begin to die, and the tumor shrinks. Now, the problem with this is that the radiation therapy, although very effective, is non-specific. So what that means is it's obviously affecting all the cells within that environment. Anything that it's hitting is going to have DNA damage. And so we often lose a lot of our immune populations in this context. And this has been demonstrated in the literature. So you can take patients that have had a radiation. Of course, you can't go around biopsying every patient's tissues, for example, but what you can do is take a blood. And you can actually see that when you survey the blood of these patients, I know this is a messy slide, but just I'll get you to focus on the arrows. What you'll notice is that in cases of, say, pancreatic cancer or even breast cancer, the number of your protective T cells in the blood diminishes. So radiation therapy is killing off these good cells. And we know that blood T cells, when they die, when exposed to radiation, if they die, we classify this as radiosensitive populations. But of course, us being TRM people, we want to know what actually happens in the tissues. I know we're taking the blood, and I know that it's very critical to understand how the immune cells respond to that. But the tissue is actually where all the action happens. It's where the tumors start. It's where the tumors grow. And so we want to know what happens to the immune populations in that site. And so we started doing some experiments where we took a model where we had some skin TRM cells as well as blood T cells, and we exposed them to irradiation. And what we found was, as previously reported, we lost our blood T cells. But what was so interesting was when we counted our number of T cells that remained, there was no difference in the skin. So something about this told us that TRM cells possess this unique ability to resist irradiation-induced death. And we refer to this as now a radioresistant population. We could also show this with using healthy human tissue. So this is both the skin and the blood. And again, we see the exact same phenotype. And so this really did pique our interest. And we were so excited because it never been reported before that there were T cells in the tissues that responded so differently than those in the blood. And how exciting is it if you can control the amount of protective T cells that could remain after this harsh therapy. And so this is when we began to try to understand the mechanisms that regulate these radioresistant properties of tissue T cells. And I've put here a really nice big Cure Cancer symbol because I want to stop and acknowledge and appreciate the fact that at this point in time, this is where the project stopped. And without the Cure Cancer funding, we actually couldn't have continued because we didn't have the means to continue. So I really, really appreciate Cure Cancer because they allowed us to continue our research into this really exciting observation. And so our first thought was, okay, well, we focused on skin and we also focused on breast cancer, but what happens to our tissue T cells and other organs? Are they also radioresistant, or is this just a skin thing? And so what we did is we took a preclinical model and we exposed it to irradiation, and then we had a look at all the T cells across a different range of sites. And I know this is a lot, but what you need to focus on is the fact that when you irradiate these populations, you can actually see that no matter where we look, whether it be in the salivary gland at the top or the liver, the gut, and the breast, you see this protection of tissue T cells. However, of course, we know that we lose the cells in the blood. So it seems to be that this TRM state, this memory T cells within the tissues, they somehow have this common feature of radioresistance. And so we wanted to look a little bit deeper and understand, well, how are they doing that? Is there some sort of DNA damage that is occurring and they're somehow protecting themselves from this? Or is there damage and then they, converting back to just a regular state? And so what we wanted to do was we wanted to have a look at this point where we could understand whether the DNA was actually broken. And so we can use a molecule called H2AX, which really detects this DNA double-stranded breaks. And so again, we took our model that had skin T cells and blood T cells, we irradiated them, and then we asked the question, do these cells have more or less of this protein called H2AX that indicates DNA damage? And you can actually see, this is kind of the raw data here on the right, but just if you focus on the plots, you can actually see there's no change in this population in the skin, but certainly in the blood, you see this increased expression of this molecule, suggesting that there's a lot more DNA damage happening in the blood T cells. But what was so critical is that the skin TRM cells weren't even being damaged by irradiation, which was really, really exciting for us. And I want to come back to understanding why we even care about this. I mean, cool story. If a T cell doesn't react to radiation, what's the point? The point is the fact that we want to know, can this be useful to us? Can we then instill it to go on and do its protective functions? And I bring you back to this, where really the purpose of a memory T cell is to give you that huge boost and rapid exposure, rapid response upon secondary exposure. And the way that it does this, so again, if you see something that's not supposed to be there, the cells can release what's called cytokines. They're like alarm signals, and we can actually measure these types of cytokines. The more you have, the better the response is. And so we wanted to understand, well, what happens when you irradiate these cells? Can they still perform their protective functions, or their functions actually? And in fact, the skin, again, absolutely, they can secrete these alarm signals, but the blood cells could not. So again, you can see that they're actually damaged. You've got DNA damage, not at the DNA level, but also at the functional level as well. And so, oops, there you go. The very last kind of set of data I'd like to present is kind of like the most exciting to us, because we took it to the next level. Okay, it's great. These cells don't die upon irradiation. They don't have DNA damage. They can still secrete cytokines, but can they prevent the growth of a tumor? That is our ultimate question here. And so what we did is we set up an experiment where we had our skin T cells, we irradiated half the cohort, and then we then challenged them with melanoma. And we said, go for it. Can you protect us against melanoma? And in fact, the case was, yes, they could. So we saw no significant difference between those TRM exposed to irradiation or not. And just to give you a clue, this is here demonstrating the tumor-free rate in the cohort. You can see that the control populations absolutely developed melanoma, whereas these guys, the mice that had TRM, they did not. And we could repeat this in a breast cancer model. So this is a triple negative breast cancer that we have in the lab. Again, we had TRM in the breast tissue. We irradiated half the cohort, challenged with breast cancer. And you can see that the tumor volume here is really low, irrespective of whether the TRM has been exposed to irradiation or not, compared to the control. So that was super, super exciting to us. We know tissue T cells are important, but now we're getting this full picture. And so just to bring it back to our summary, hopefully I've convinced you that TRM cells or T cells are critical for fighting cancer. We know that the tissue T cells, the actual ones in the tissue, not in the blood, they can survive irradiation and they can still protect against tumors. This feature of radioresistance seems to be common amongst all cells that reside in the tissues, and they don't seem to exhibit DNA damage, and they still retain their functional capacity. And so now what we're trying to understand is, well, what's the molecular basis for conferring this? How can we package this, or how can we use this information to develop some sort of drug or therapy that allows all the other good immune cells to remain protected and to continue on with their functionality in the face of harsh irradiation? And so what we aim to do is develop some sort of therapy that allows us to retain our T cells so we can get better immune protection. So with that, I'd like to end by thanking my supervisor and mentor, Professor Laura McKay at the Doherty, to all the team members, to our funding bodies, but of course, the biggest thank you to all the donors and fundraisers that contribute to Cure Cancer, that allow us to do what we actually do here. And of course, the consumer representatives who without them don't motivate us to really get out of bed and try as hard as we can to fight this thing. So with that, I'll stop. Thank you. And I'm happy to take any questions.